Good morning, everyone. I am delighted to welcome you all today. I am Dr. S.K. Singh, and I hold a PhD in this field. Today, I will be discussing the analysis of food colors used in formulations. In this presentation, we will explore the different types of food colors, their applications, and how they are analyzed for quality and safety. I look forward to sharing insights and engaging with you throughout this session. Let's get started. Today, we'll be looking at the analysis of food colors used in pharmaceutical and nutraceutical formulations, guided by standards from FSSAI, USB, EP, WHO, and Codex guidelines. This is applicable not only to pharmaceuticals, but also to nutraceuticals, food, and dietary supplements. Food colors are added to these formulations for several key reasons. First, they help improve the aesthetic appeal, making the product more attractive and appealing to consumers. Second, they are used to maintain batch-to-batch uniformity, ensuring consistency in every production cycle. Third, they play a role in enhancing patient and consumer acceptance, as people often associate certain colors with specific benefits or qualities. However, it's important to remember that not all colors are safe or compliant. Uncontrolled or non-compliant coloring agents can pose serious risks, such as toxicity, which can harm health. They can also lead to regulatory noncompliance, resulting in legal issues and potential product recalls. So, ensuring the safe and compliant use of colors is crucial for maintaining product safety, quality, and trust. This highlights the importance of adhering to the guidelines and standards set by these authoritative bodies, which is a key part of our overall focus on quality and safety in formulation development. Let's take a look at the major regulatory authorities that oversee food and color safety around the world. First, we have the FSSAI in India, which enforces the food safety and standards regulations. This body ensures that food products, including color additives, meet strict safety standards within India. Next is the USB, NF, or the United States Pharmacopoeia, National Formulary, which provides monographs specifically for color additives used in pharmaceuticals and food. It's a key reference for ensuring quality and safety. Then we have the European Pharmacopoeia, or EP, which sets quality standards for medicines and related substances, including color additives, across Europe. The WHO and FAO, through the Codex Alimentarius, develop international food standards, guidelines, and codes of practice. Their work helps harmonize safety standards globally, making sure color additives are safe and properly regulated. Finally, in the United States, the FDA's CFR Title 21 is the regulation that governs food additives, including colors. It specifies which colors are permitted and under what conditions. An important point to remember is that only those colors that are permitted within these regulatory frameworks and within defined limits are allowed for use. This ensures consumer safety and product consistency across markets. So, throughout this presentation, keep in mind that adherence to these authorities and regulations is crucial for safe and compliant use of color additives. Now let's take a closer look at how food colors are classified. First, we have natural colors. These are derived from natural sources like plants and other natural substances. For example, curcumin, which gives that vibrant yellow color, and caramel, often used for browning or coloring baked goods. Then there's chlorophyll, the green pigment found in plants, and beetroot red, which imparts a deep red hue. These natural options are often preferred for their perceived health benefits and minimal processing. On the other hand, we have synthetic or permitted colors. These are artificially produced but approved for use in foods. Some common ones include tartrazine, known as E102, which provides a bright yellow color. Sunset yellow FCF, or E110, is used to give a warm orange tone. Carmazine E122 adds a reddish hue, and brilliant blue FCF, E133, gives a vivid blue shade. These synthetic colors are widely used because they are stable, vibrant, and cost-effective. Understanding these classifications helps us grasp the broader theme of food additives and their safety and transparency. Whether natural or synthetic, the choice of color impacts not just appearance, but also consumer perception and health considerations. Let's take a closer look at the regulatory permitted limits set by FSSAI. When it comes to synthetic colors, the maximum limit is typically up to 100 parts per million, whether these dyes are used alone or in combination. This means that any food product should not contain synthetic colors exceeding that concentration to stay within the safety standards. For pharmaceutical preparations, the limits can vary depending on the specific product, so they are governed by product-specific regulations. It's important to follow those guidelines carefully to ensure safety and compliance. Now, an essential point to remember is that certain dyes, like metanyl yellow and Sudan dyes, are strictly prohibited. These non-permitted dyes are considered unsafe, and their use is completely forbidden by the authorities. Connecting this to our overall theme, regulatory limits like these are crucial for ensuring food safety and protecting consumer health. It's vital for manufacturers and regulators to adhere strictly to these limits and bans to maintain trust and safety in the food supply. Let's take a closer look at the quality attributes of food colors. When we talk about critical parameters, the first one is identity. It's essential to confirm that the color is the correct compound and matches what we're expecting. Next is color strength, which refers to the intensity or vividness of the color, something that directly impacts the visual appeal of the food product. Purity is also vital. We need to ensure that the color is free from contaminants and impurities that could affect safety or quality. Heavy metals are another concern. These are toxic elements that can sometimes be present in pigments, so testing for them is crucial for consumer safety. Organic impurities and residual solvents are also important parameters. They help us verify that no unwanted organic compounds or leftover solvents remain from manufacturing processes. Lastly, microbial quality is critical where applicable, especially in products that are more prone to microbial contamination, to make sure the colors are microbiologically safe. All these parameters together help us ensure that food colors are safe, effective, and of high quality, integral to maintaining consumer trust and product integrity throughout the supply chain. Let's talk about sampling and sample preparation. First, it's crucial to take a representative sample from the finished formulation. This ensures that our analysis accurately reflects the entire batch. Next, we need to protect the sample from light and heat, as these factors can alter the sample's composition and affect our results. When it's time to prepare the sample, we dissolve it in a suitable solvent, such as water or methanol, depending on the nature of the compound we're analyzing. After dissolution, filtering the sample through a 0.45 micron membrane is essential to remove any particulates that could interfere with our analysis. Finally, we prepare both working standard solutions and sample solutions, which are used in our analytical procedures. Proper sampling and preparation are key steps to ensure accuracy, precision, and reproducibility in our results, tying directly into the overall quality of our analytical process. Now, let's talk about acceptance. The lambda max, or the maximum absorption wavelength, should match the standard within a margin of plus or minus 2 nanometers. This is a crucial point because it helps us confirm the identity and purity of our substance. When conducting identification tests, one common method we use is UV, visible spectrophotometry. This technique allows us to analyze the sample's absorption spectrum and identify characteristic peaks. To verify the identity, we compare the lambda max of our sample with that of a reference standard. If they match within the acceptable range, it indicates that our sample is consistent with the standard, which is a good sign of quality and correctness. Remember, this comparison is key to ensuring the reliability of our results and maintaining the integrity of the testing process. Now, let's look at the assay by UV visible method. The principle behind this technique relies on the Beer-Lambert law, which states that the absorbance of a color solution is directly proportional to its concentration. So, essentially, the more concentrated the solution, the more light it absorbs, which we can measure accurately. When it comes to calculation, we use this formula. Assay percentage equals the absorbance of the sample divided by the absorbance of the standard, multiplied by the standard weight over the sample weight, and then multiplied by 100 to get a percentage. This helps us determine how much of the active compound is present in our sample compared to a standard. It's also important to note the typical acceptance range for this assay, which is between 85% and 115% of the labeled color content. This range ensures that the product meets quality standards and is within acceptable limits for potency and safety. By understanding these key points, we can effectively apply UV visible spectrophotometry to ensure our products are consistent and meet regulatory requirements. Now, let's take a closer look at the assay by HPLC method and the chromatographic conditions used as an example. When we talk about this technique, the choice of the column is crucial. In this case, we're using a C18 column, which measures 250 millimeters in length and 4.6 millimeters in diameter, with a particle size of 5 micrometers. This type of column is popular because it provides good separation and is suitable for a wide range of compounds. Next, the mobile phase typically consists of a buffer and methanol. The buffer helps maintain the pH and ensures reproducibility, while methanol acts as the organic solvent to assist in eluting the compounds from the column. The flow rate here is set at 1 milliliter per minute, which balances between efficient separation and reasonable analysis time. For detection, we're using UV or PDA, which is common in HPLC because many compounds absorb UV light, making it easier to identify and quantify them. We inject 20 microliters of sample into the system, a volume that provides a good balance between sensitivity and peak shape. Now, why do we use this setup? The main purpose is quantification, accurately measuring how much of a substance is present. Additionally, this method is effective for separating mixed colors, which can be important in quality control or identifying different components in a mixture. Overall, these conditions are carefully chosen to optimize the separation and detection of compounds, ensuring reliable and reproducible results in our analysis. Let's take a look at the limits for organic impurities as outlined by USB and EP requirements. These standards are essential because they help ensure the safety and quality of pharmaceutical products. When we talk about limits, we're referring to the maximum allowable levels of these impurities in our drug substances or finished products. It's important to remember that these limits are set based on toxicological data and the potential risks associated with each impurity. By adhering to these guidelines, we can maintain product integrity and protect patient health. Overall, understanding and controlling organic impurities is a key part of our quality assurance process, and it ties directly into our broader goal of compliance and safety in pharmaceutical manufacturing. Now, let's take a closer look at the methods we use for testing heavy metals and toxic elements. We primarily rely on two sophisticated techniques: ICP-MS, which stands for Inductively Coupled Plasma Mass Spectrometry, and AAS, or Atomic Absorption Spectroscopy. These methods are highly sensitive and accurate, allowing us to detect trace levels of metals that could pose health risks. Moving on to the key elements we focus on, heavy metals like lead, mercury, cadmium, and arsenic are the main concerns because of their toxicity and potential to accumulate in the body. We also look for other toxic elements that might be present. When it comes to testing, we establish specific limits. These are safety thresholds set by regulatory agencies. Our goal is to ensure that the levels of heavy metals and toxic elements in a product or sample stay well below these limits, guaranteeing safety and compliance. Overall, this testing process is crucial in our comprehensive approach to quality and safety assurance. It helps us identify potential risks early and maintain the integrity of our products, ultimately protecting consumers and supporting our commitment to excellence. Now, let's discuss some prohibited dye testing, which is crucial for maintaining food safety standards. The target dyes we focus on include metanyl yellow, Sudan I through IV, and rhodamine B. These dyes are often used illegally or inappropriately, so detecting their presence is important. The methods we rely on for detection are TLC, HPLC, and LCMS, which are highly sensitive analytical techniques. The key requirement here is that the dye levels should be below the detection limit of these methods, ensuring that no harmful dyes are present in the food products we examine. This helps us ensure consumer safety and compliance with regulations, reinforcing the importance of strict testing protocols in our overall food safety framework. Now, let's look at an example calculation from the UV assay. The absorbance of our standard is 0.52, and the absorbance of our sample is 0.498. To determine the assay result, we divide the sample absorbance by the standard absorbance, which gives us 0.498 divided by 0.52. Then, we multiply that ratio by 100 to express it as a percentage. Doing that calculation, we arrive at approximately 95.77%. The key point here is that this result indicates our sample's concentration relative to the standard. Since it's close to 100%, it suggests that our sample is within acceptable limits. And importantly, this means our sample complies with the specifications we set. This example highlights how straightforward calculations like this help us verify the quality and consistency of our samples, which ties back into the overall theme of ensuring accuracy and reliability in our analytical methods. Let's wrap up with some key takeaways. First, food colors need to be permitted, pure, and used within established limits to ensure safety. Using approved colors in the right amounts is crucial for consumer health. Second, tools like UV, vis spectroscopy, and HPLC are our primary analytical methods. They help us verify the purity and concentration of colors, ensuring quality control. Third, testing for heavy metals and prohibited dyes is mandatory. These tests help us prevent harmful substances from reaching consumers. Ultimately, compliance with regulations not only protects consumers, but also ensures our products are accepted in the market. Remember, color may attract the eye, but compliance is what builds true trust with our customers. For further support, please feel free to contact us. We're here to help clarify any questions you might have or provide additional resources. Remember, the goal is to ensure you feel confident moving forward. Thank you so much for your time and attention today. We truly appreciate your engagement and look forward to supporting you in your next steps.